So today inshallah we will start our first exercise, and this first exercise it is related with measurement of the fasting and random blood glucose level. How we will measure this glucose level. Before going into the detail of the procedural part, let's first have a brief idea or introduction about how the glucose level is maintained in our body. In relation to blood glucose level, two terminologies are in common use. One is called the fasting blood sugar or FBS, and second one is random blood sugar or RBS. Fasting blood sugar means level of glucose, level of sugar in the blood that is measured after a period of 12 hours fasting. And 12 hours fasting means that when the person is not allowed to eat or drink anything which contains some energy for a period of 12 hours. And here, restriction of the energy intake is not only through the oral route, by any other means as well. No injections, no drips, nothing. But during this time, if he has a desire or he feels thirsty, then he can take some quantity of water. So some quantity of water, not too much water, but some quantity of water you can allow, but not the other. Normally, fasting blood sugar level varies between 80 to 100 milligram per year. 80 to 100 milligram per year. But these values, they are variable from area to area and lab to lab. Some labs consider the value up to 110, some labs up to 120, and some labs up to 125. But 125 is the maximum limit or the last limit for this fasting sugar. Similarly, the blood sugar level measured without any particular time, or in other words you can say which is measured at any time after taking food. Maybe after 2 hours, maybe after 1 hour, maybe 6 hours, maybe 8 hours. Any time, whenever you want, you collect the blood sample and measure the blood sugar level This is called random blood sugar level. And normally the random blood sugar level, it must be less than 140 milligram per dl. Less than 140 milligram per dl. In addition to these two terms, that is fasting and random, another term is now in common use, which is called postprandial blood sugar level. Postprandial. Postprandial means prandial, P-A-N-D-R-I-A-L, prandial. Postprandial means that you ask the person to come with 12 hours fasting. With 12 hours fasting. Collect the fasting blood sample and then you ask him to take his breakfast and then again come back about 1.5 to 2 hours after taking the breakfast. And then after this 1.5 to 2 hours, you have to collect another blood sample to check the blood glucose level. This blood glucose level, it will be called postprandial. And why this is important? Because we believe that in a normal person, the blood sugar level that increases by taking of the food, it has to come back to the baseline by 1.5 to 2 hours. So by taking this blood sample, we can have an idea that in this person how much blood sugar level rises and whether it comes back to the normal within this time or still it is at a higher level. After 1.5 to 2 hours, if the blood sugar remains still at a very high level, it means that the patient has some problem with the insulin secretion. So that's why nowadays this postprandial blood sugar level measurement is an important diagnostic part. Now let's have an idea that how this blood sugar level is maintained and what is the need of this maintenance. Why this blood sugar has to be maintained. The first part, that why it has to be maintained. There are some tissues in our body, some organs in our body which depends for their energy supply totally on glucose. Means they will not use energy from any other source like fats or proteins. They need only glucose for their energy. Like number 1, brain. Number 2, retina. Number 3, germinal epithelial cells. So all these structures and many more, they depend totally on glucose for their energy supply. And therefore, a blood glucose level has to be maintained within a normal range. Otherwise, these tissues cannot get the proper energy supply and maybe they will stop working. So that's why in our body, irrespective of the point that we are taking the food or we are not taking the food, the blood glucose level has to be maintained. For example, during Ramadan, we are not taking the food for a period of 14 hours, 15 hours, 16 hours like this. But still we are able to do all the activities because the blood glucose level is kept maintained. Now how this is maintained? This is maintained by the opposite effects of two hormones called insulin and glucagon. Insulin and glucagon. These two hormones, they are involved in regulation of blood glucose level with producing opposite effects. Insulin is said to be a hypoglycemic hormone. Hypoglycemic yani it is reducing the blood glucose level, decreasing the glucose level. Whereas glucagon is a hyperglycemic hormone, increasing the glucose level. How the insulin will decrease the glucose? Whenever the blood glucose level goes more than the normal values, pancreas starts secreting more and more insulin. And this insulin, it will act on the cells and increases their permeability for the passage, for the entry of glucose inside the cells. So it is the result of increased permeability of the cells by the insulin that the glucose can get inside the cells. And as more and more glucose is going inside the cells, the blood glucose level will come down. But along with this insulin mechanism, there is one more mechanism in our body where the glucose can enter even without insulin. And that mechanism, it is restricted, it is available only in our skeletal muscles. So skeletal muscles during the activity period, during exercise period, physical activity, they are capable of taking the glucose even without the presence of insulin. This is the reason that for every diabetic, we advise them to have a physical activity daily at least for half an hour, so that all the extra glucose, it can go inside the muscles. Similarly, glucagon, a hyperglycemic hormone that is increasing the glucose level, it can produce this increase by two different mechanisms. Number one, by the process of glycogenolysis, breaking down the glycogen. But this breaking down of the glycogen is available only from the liver cells. The muscle glycogen cannot come out once it is inside the muscle. It can be used by the muscle tissue, but not available for the other tissues. So, under the influence of glucagon, the liver glycogen is broken down, and so more and more glucose is available in the blood. Number two, under the influence of glucagon, the process of gluconeogenesis will be stimulated, which means formation of glucose from substances other than carbohydrate, that is fats and proteins. So glucagon, using fat and protein, synthesizes the glucose. So by these two mechanisms, glucagon try to bring the blood glucose level back towards the normal range whenever glucose level goes down. And whenever the hypoglycemia appears, glucose level goes down the normal below 80 milligram, insulin secretion will decrease, glucagon secretion will increase. Whenever hyperglycemia develops more than 100, 110, 120, glucagon secretion will decrease, insulin secretion will increase. So this feedback system between the insulin and the glucagon, it is responsible to regulate or to control the blood glucose level in our body normally. And if the system is disturbed because of deficiency of insulin or defective quality of the insulin or because of defect in the receptors for the insulin, anything, then the condition will appear, which is called diabetes, characterized by a hyperglycemia and inability of the cells to utilize glucose. Now, after this, a brief introduction about the glucose regulation. Let's see how we can measure this glucose in our exercise. The first thing is that here we will restrict our measurement only for the random blood sugar level, because now at this time a fasting sample is not available, so we will restrict only for measurement to the random blood sugar level. This measurement, it consists of 4 different steps. The first step of the procedure is to collect the blood sample and to get serum or plasma from it by the process of centrifugation. What we will do, we will take human blood sample and then put the sample in this tube, in this bottle, which is a heparinized container. Inside walls of this bottle, they are coated with heparin. So because of the presence of heparin coating, the blood will not clot. Then what we will do, we will transfer the blood sample in many of these small tubes called Eppendorf tube. Its name is Eppendorf tube. Eppendorf, Eppendorf tube. And these Eppendorf tubes, they are used to collect any of the sample, whether blood, whether urine, whether saliva, whatever it may be. And also you can use these Eppendorf tubes for centrifugation purposes. And the facility is that you can close it as well. Now, what we will do, we will transfer our blood sample in these Eppendorf tubes, put the Eppendorf tubes here in this small centrifuge, and then the process of centrifugation will be performed, and as a result, we will get this plasma, this clear fluid, yellow colored from the blood. The blood cellular part is separated out, the large protein separated out, and only the fluid part is collected, which is called plasma. Why this is called plasma? This is not serum, this is plasma. Why this is plasma? Because we have used for centrifugation a non-clotted blood sample. Non-clotted blood. So this contains all the components of the plasma, including the clotting factors. But if you will not use heparinized tube, if you use an ordinary tube, then the blood will get clotted in it. And when the clotted blood will be centrifuged, you can get a similar type of solution, but that will be called serum, not plasma. Now what is the difference between plasma and serum? Only one difference, that is of clotting factors. In plasma, clotting factors will be present. In serum, clotting factors will be absent because they are used for clotting of the blood. So except this one difference, serum and plasma has same composition, no difference, excepting the presence or absence of clotting. So remember this difference. If you are asked that what is the difference between serum and plasma, you should to say only the difference of clotting factors. Serum, clotting factors absent, plasma, clotting factors present. So this plasma we will collect here by centrifugation of our blood sample. Then the second step, here you will take 2 test tubes. One test tube you mark T for test, and the second test tube mark S for standard. So T for test and S for standard. Then next step, you have to take a chemical kit used for glucose analysis. And this chemical kit, it contains 2 bottles, a big bottle and a small bottle. This big bottle, it contains a chemical solution which contains some enzymes, a buffering material, as well as a coloring material. And this chemical we will mix with our sample and with our standard to develop a reaction between it and the glucose and appearance of a pink color. The glucose in our sample and in our standard, it will react with the chemicals of this solution, and as a result of this reaction, a pink color will appear, a pink color will develop.
What's the name of this?
This glucose analyzing kit. Yeah, glucose measurement kit. Anything you can say. The second bottle, this small one, it contains the standard glucose solution. Any solution is said to be standard when you know the amount of particular substance in that solution. What is the amount? So then you can say it is standard. So here it is standard glucose. Why? Because we know that in this solution glucose is present in a concentration of 100 milligram per dl. So glucose concentration here in this is 100 milligram per dl. So that's why, due to known concentration, this solution is called standard glucose solution. And it is used for comparison. With this, we will compare our sample. So in the next step, after taking the 2 test tubes, what we will do, we will take this glucose measuring chemical or kit and add 1 ml. How much? 1 ml or 1000 microliter. 1000 microliter is equal to 1 ml. So 1000 microliter or 1 ml you will add in both the test tubes, that is test tube T and test tube S. In the next step, you will take your plasma and now add 10 microliter. How much? 10 microliter of this plasma in the test tube T. Only test tube T. Whereas in the test tube S, you will add 10 microliters of this standard glucose solution. 1 ml of glucose analyzing kit in both T and S, no difference. But secondly, 10 microliter of glucose standard in the test tube S. 10 microliters of plasma in the test tube T. Then you mix the solutions by shaking the test tubes and place them in the water bath for 10 minutes at 37 degrees centigrade temperature. Test tube T, plasma 10 microliters. Test tube S, standard glucose solution, also 10 microliters. This is a standard glucose solution. 10 microliter. From here you will add in test tube S. This is plasma. 10 microliter. From here you will add in test tube T. But this solution 1 ml in both.
Then after the 10 micro,
Mix them and then put them in water bath for 10 minutes. Now in the water bath, you have another option that in place of 37 degrees centigrade, you can maintain the temperature at 20 to 25 degrees centigrade also. But in that case, the incubation time has to be increased. Then in place of 10 minutes, you have to put for 15 to 20 minutes. So better to put 37 degrees centigrade and for 10 minutes. Now after 10-minute incubation, your preparation of the sample is now complete. Now you go for the third step, which is called spectrophotometry. Spectrophotometry is the process in which we will put our samples in this machine called spectrophotometer and passes through the sample a particular wavelength of light. And then we will measure that how much amount of light is absorbed or how much amount of light passes away. Anything you can measure. Either amount of light absorbed or either amount of light that passes away. Both things you can measure. And then we will compare that how much amount of light is absorbed by sample and how much amount by our standard. And then in the fourth step, we will use these values to calculate the glucose level milligram per ml by using a formula. For the purpose of spectrophotometry, first you have to prepare your spectrophotometer. How to prepare the spectrophotometer? Number one, depending upon that how much wavelength of light you are using, you have two options in your spectrophotometer. One option is called UV, ultraviolet, and the other option is called VIS, visual, from visual VIS. Any one option you have to select. whether UV or whether VIS. Now what will be the criteria, how you will select? The simple method is that if the wavelength of light that you are using is 500 or more than 500, 500 or 500 plus, 510, 520, 540, 650, 730, whatever it is. Then in this case, you will choose UV. So 500 or more than 500, you select the option of UV. Less than 500, 350, 380, 400, 450, VIS. So very easy. Just remember 500 or more UV, less than 500 VIS. Secondly, you will observe, you will find a knob on the top of the spectrophotometer. So by adjusting, by rotating this knob, you have to adjust your required wavelength. Whatever wavelength you want, adjust it. Third step, in the spectrophotometer, you will have different options like absorbance, transmittance, concentration, multiplication factor, so many options. What option you want to measure, you have to select. Usually, we select the option of absorbance, that is how much light is absorbed. And absorbance is designated by the sign triangle. Triangle means absorbance. So whatever the option you want, you can select. Usually in our practice, we select the absorbance. Then finally, the fourth step is zeroing the machine, making the machine zero. For this purpose, you can use either distilled water or you can use a solution called blank solution, B-L-A-N-K, blank. So, according to the recommendations of the company, sometimes you use blank solution, sometimes you use distilled water to make the machine zero. Once you choose all these four steps, now it means that your spectrophotometer is ready. You can put your sample and standard there and measure their absorbances. Another thing here is that in order to measure your sample and standard in the spectrophotometer, you need a specific tube called the cuvette. Its name is cuvette. This one. Two types of cuvettes we have. There are some important precautionary points for using the cuvette. Number one, the cuvette should be clean and dry. If it is not clean, you can wash it, clean it, and then dry it by the tissue or piece of cloth, whatever. So dry cuvette should be used. Number two, the minimum quantity of solution you have to put in the cuvette is 1 ml. Minimum 1 ml should be there. Otherwise, if the solution is less than 1 ml, your results will not be accurate. So for accurate results, 1 ml is the minimum quantity in a cuvette. Thirdly, for all the samples and the standard, use the same side to put in front of the light. Here in this cuvette, all the four sides, they are similar, so no problem. Any place, any side, any direction can be used. But there are other cuvettes which have two sides different. In that case, you have to remember that the same site for all samples should be used. Okay? So these precautions you have to remember for your proper results. Also, whether it is the cuvette or whether it is the test tube, you should to mark them properly, which is sample, which is standard. If more than one sample, then sample number 1, 2, 3, 4, so that you will not mix them. Otherwise, later on you will forget which one was standard, which one was sample. And then you will not found the proper results. So that's why everything should be done carefully. After the spectrophotometry, here in case of glucose measurement, the wavelength that we will use will be 540 nanometers. So adjust your spectrophotometer to 540 nanometer wavelength and measure the absorbance of your sample that you prepared, that is test solution, and the absorbance of your standard solution. Once you have measured the absorbances, now you have to calculate the glucose or the random blood glucose level milligram per dl by using this formula, that is absorbance of T divided by absorbance of standard multiplied by concentration of standard, and concentration of standard here in this case is 100. So absorbance of test divided by absorbance of standard multiplied by concentration of standard. This is the formula to calculate glucose level milligram per dl. The units are must. Your result, if you write without units, it will not be considered correct. So remember, very important point, that every time when you write any result, not only for the glucose, for anything, even for blood pressure, even for heart rate, whatever, the units are lazim, you should to write the unit, which unit you use. Okay? Now, these are the glucose measuring solution. This is the standard solution. This is the plasma samples available. So micropipettes and everything you need available on the back bench. Prepare and remember, two things very important for you. Number one, for every exercise, prepare at least 3-4 samples, so that if some of them are wrong, some of them are correct, and it will be suitable for you. Secondly, you have to perform these experiments in the exam as well. So until and unless you perform it during the classes, you cannot do in your exam. So that's why instead of passing your time in chatting, laughing, talking, playing mobiles, perform the exercise. It is hardly 15-20 minutes work, so just perform it, spend this time in learning. After finishing, you can have enough time for your entertainment. So please don't waste your